Okay, thanks, Rick. So today, in my talk, I'd like to sort of provide an overview or summary of the standard methods that are being used for dynamic functional connectivity characterization. And one of the goals is to, cover some of the topics that will be covered in some of the later scientific abstracts. No disclosure, conflicts to declare. So this is the outline. So we're going to start off by very briefly making the distinction between static and dynamic functional connectivity, and then briefly describe the most basic form of dynamic functional connectivity, which is sliding window correlation. Then we'll go on to sort of more different variations of dynamic functional connectivity. So most of you are familiar with resting state fMRI, in which we look at the resting state fluctuations, and we look at signals from different regions of interest. We can correlate them, and typically that correlation is done over the entire scan period, whether it's 5 minutes or 10 minutes or 15 minutes, and we can come up with a correlation matrix here. So this would be, for example, a static functional connectivity correlation matrix. For dynamic functional connectivity, the most basic form is what's called sliding window correlation analysis, in which we simply take windows and do the correlation in each window, and we slide that window along. And as we do the sliding, we record the functional connectivity in each window, and we plot this functional connectivity as a function of time, or in this case, a sliding window number. The time courses you use can be based on different parcellations. So for example, you might use an anatomic parcellation. You might use one from, say, the Human Connectome project, or you might do ICA on your data and come up with a parcellation based on that. But in any case, that step is, there are different variations for that, but at the end, you are then essentially performing sliding window correlations of the time courses from various regions of interest. So let's talk a little bit about some of the things that can go wrong with DFC and some of the issues with DFC, which is sort of short for dynamic functional connectivity. One thing is there's a lot of variation in terms of what window length to use. It's not clear, it's not totally obvious at the outset what window length you would use. One thing to keep in mind, though, is that if you simply have random data and you correlate it and do sliding window correlation, you will end up with sort of dynamic functional connectivity time series that sort of look like there might be some information there, but that's just simply due to random fluctuations. And those random fluctuations, sort of, the size of them is sort of proportional to 2 over the square root of the window length. So if you have a very short window, so for example, in this case, a 15-second window, you see a lot of sort of variations as compared to, say, the 90-second window, where it's fairly close to this magenta line, which is the truth. Basically, these two waveforms are uncorrelated over time, and yet you might conclude that there is dynamic functional connectivity when you use a small window. So you do need to be careful about not using too small of a window. Also, the other thing to note is when you do these estimates, because you're doing a sliding window, depending on how quickly you advance that window, the neighboring estimates can be highly correlated, which makes sense because basically you're as you're moving the window along, you're dropping one data point, but then you're getting the next data point, and there's a lot of sort of shared data points between windows. And so you sort of see that, it's not like this waveform can just change as rapidly as possible. There is sort of this smoothness to that because you're sliding the window, okay? So what is the recommendation on what to do? Well, the recommendations are to, first of all, keep the window length greater than 1 over the minimum frequency. The idea is if you have say, a sinusoid, the minimum window should be at least as greater than the period of that sinusoid. So for example, if your minimum frequency is 0.01 hertz, then you need a window greater than 100 seconds, okay? So for example, here we're showing, we assumed we had sort of frequency of 0.025 hertz, which could correspond to 40-second window length. And you can see when you're less than 40 seconds, there's just a lot of variation in the estimates, okay? And then as you get above that, it sort of diminishes. And so the recommendation is to use high-pass filtering, so you can set your minimum frequency, and then based on that, you choose your minimum window length. The other thing to note is that, so that's on the short end. On the long end, obviously, as you pick longer and longer windows, you're starting to smooth out or filter out the fluctuations of interest. So for example, in this plot here, you see that with 32-second time window, you're seeing fluctuations here, which are quite strongly damped out once you go to 128-second window here, okay? So there are some, so there are sort of this understanding that there is an effective window length, but really there is really no consensus at this point as to what window length to use. I would say in the literature, this is from a review article by Priti and others, window lengths typically lie in the range from 30 seconds to 60 seconds. And it just so happens that the most common window length is about 20 TRs, okay, which is about 40 seconds to 60 seconds given a TR of, say, 2. Now, obviously, if you're doing multi-band, then that would be probably more TRs. So we've talked a little about some of the sliding window DFC issues. Some other issues are the simplest case is to use a rectangular window, but people have also proposed using tapered windows, which sort of are more smoothly varying in the sense that they can be less sensitive to sort of transitions between the time series. And then you also have to sort of choose how the window offset or the step size. So, in what step do you advance the window? And the most common step sizes are either you advance the window with step size equal to the window size, which is basically you have non-overlapping windows. And the other choice is a step size of 1 TR, which is essentially you just advance the window the minimum amount. But there really is a lack of consensus there. So for example, this is a histogram of number of articles versus window size, and you can see people either pick this one or this one for no really good reason, but just to keep in mind. So there's still a lot that is not really standardized in this area. So I think definitely more work should be done here. So now we're going to talk about, so one of the issues with sliding window correlation is you need to pick the window size, and one way of getting around that, and which we'll be discussing in one of the scientific abstracts, is a technique called dynamic conditional correlation from Martin Lindquist's group at Johns Hopkins. So this is a slide that we saw before where this is a truth, and so there's no variation in correlation over the time series. And as you remember, as we use a very short window size, we get lots of variations, and it reduces as we use longer window sizes. So dynamic correlation is a method that comes out of the finance literature where people are very interested in predicting stocks and what's going to happen with the economy. And so using this method, they find that you can actually get much, much closer to the ground truth in here. So this is the DCC sort of estimate of what the correlation is between two time series, and it matches the truth fairly well. So I'm not going to go through the details, but let me just walk you through what DCC does. So it is a form that came out of actually Bob Engel from UCSD and later NYU, I believe, won the Nobel Prize for this Garch model, which allows for time-varying variance in your time series. So this is an example of a time series where the idea is the variance is clearly increasing here and here, and the idea is you want to update your variance estimate in your time series. And so that's important if you're going to capture what's going on in the data. If you just assume that this had the same variance across time, you'd be making the incorrect assumption and not be able to predict what's going on. Then the next thing they do is they assume that there is also this update rule for the covariance matrix. And so what they're doing is they're predicting the covariance matrix with time. So at every time step, you're updating this estimate of the covariance matrix, which is essentially the correlation between the two time series. And so that's the basic idea that you have, and that current estimate is based on a weighted average of the previous estimate and an update based on the current data. And this has some nice theoretical properties, which you can read about more if you'd like to in the Lindquist paper. But one idea is that it seems to, at least in simulations, do much better at capturing ground truth. So for example, here, this dotted line here shows the ground truth. And here we have the ground truth here, this sort of variation, sinusoidal variation. And these are different methods. And the DCC method here sort of follows most closely along with that. And that's shown by the mean squared error here, where of all the different methods, it seems to do much better than these sliding window methods. Now, I must say the DCC has not been really widely adopted yet, but it's probably something that we need to consider looking at in some more detail. And one of the abstracts will be using that as well. Okay, so once you have the dynamic functional connectivity time series, you can do various different things on it. For example, you could look at the standard deviation of it. You can look at different frequency contents of it. So there's many things you can do with a time series, and so anything you can think of doing with a time series, you can do with the DFC time series. So I'm going to focus on one thing that's been quite popular in the area, which is characterizing what states there might be in the brain from the DFC estimates. And it turns out there's really two sort of potentially complementary but not equivalent ways of doing that. One is characterizing the states directly from the DFC estimates, which I'll talk about first, and then we'll talk about direct data modeling next. So what do we mean by characterizing the states? Well, imagine that this is the dynamic DFC between sort of two time series, and obviously if you did it between every set of pairs, you would end up with these correlation matrices that vary with time, and so that's essentially the data you're dealing with, this 4D, in this case, this 3D data set. And the idea is from here, you want to extract what states potentially best represent this data. So the idea is maybe this data can be represented in a lower dimension that might represent different brain states. There's different ways of doing this. One of the first ways that was proposed was you take these correlation matrices and you might even aggregate them across subjects. And then you could perform, for example, a k-means clustering, in which case you're trying to sort of sort these into areas. So this is a geometric picture of k-means clustering. In some high dimension, you're trying to find those divisions of the data in which there's a clustering of the data into non-overlapping areas here. So this is an example from Vince Calhoun's group where they did that clustering and they identified 5 different states and they compared it between healthy controls and also, so these are the states in the healthy controls. And what they could do then is they could, so for each state, there's a different connectivity matrix. And then you can start asking questions like, well, how often does the brain spend in each state? How does the brain transition between one state and the next? And so, for example, in this study here, they basically looked at the mean dwell time, or how often the brain stayed in a different state. And they found that in healthy controls, there was more staying in states 1 and 2. Whereas in the schizophrenic population, they were more in stages 4 and 5. Okay. This is a study using more of a task-based paradigm from Javier Gonzalez Castillo, where they are giving different sort of tasks, and so you're actually driving the brain into different functional connectivity states. And once again, k-means clustering, and they were able to show that from that you could identify what state the brain was in a case where you're actually driving it. Okay. So there's this idea that, so the basic idea is you take the DFC data and you're doing some higher order clustering of the data. So that's one approach. But the other thing is you could say, well, instead of going through this process and calculating these correlation matrices, I could actually just go to the data itself and see if there's any clustering in the data. And this is more along the lines of state modeling. And then once I have that, then I can apply the same analysis I would have done to these states. But now the way I've gotten the states is quite different. Where this really, one of the earliest examples of this was from Xiao Lu and Jeff Dunn, where they noticed that there were these reoccurring patterns of sort of coactivation patterns in the data. And in this case, they simply k-means cluster the data itself, and they find these different coactivation patterns. Okay? Now, you can get a little more sophisticated and do what's called hidden Markov modeling, in which you assume that at every time point, your data comes from a hidden sequence where each state has a mean activation and connectivity pattern, okay? And then is essentially going between the different states with some probability, and you're trying to estimate what the brain is doing in time directly from the data itself without computing any connectivity to begin with. So this is an example from a recent PNAS paper where they applied hidden Markov modeling to resting state fMRI data. And so these are some of the states they found. So once again, they're saying the claim is that every time point in the fMRI time series is a realization of one of these states, where each state has a mean activation pattern and then also a covariance matrix, which is essentially the functional connectivity inherent to that state. And so for every time point, you can, since it's a probabilistic framework, you can say, what is the probability that I was in that state? And you can assign sort of different states. And then you can also look at the transition probability between states. And this is a very interesting study in the sense that they found that here they have 12 different states, and they found out that there were these two metastates where the brain would transition between this cluster of states or preferentially stay in this cluster of states, but once in a while make a transition between these two different states. So then the question is, what's the right way to analyze the brain? So just to review, we have, for example, we could sliding window analysis, do k-means clustering. We could end up with these states and ask questions about mean dwell times, fractional occupancy, which is, how often are you in a state, transition probabilities between states, or you could do hidden Markov modeling, where you end up with a different set of states and you end up with different dwell times. So the answer is not yet clear, and it's up to people in this audience to help answer that question. But one thing that was pointed out in this recent paper by Nielsen and others is that what assumptions you make in your analysis have a fairly big impact on what claims you might make. For example, here, if you do hidden Markov modeling where there's no fixed window size and the data sort of determines the transitions, then you can sort of see there's a lot more variability in the state lifetimes, which is how often it spends in each state, versus here in the windowed mean case clustering, where there's a fairly more homogeneous distribution of states, which sort of makes sense because you're using a windowed correlation with a fixed window size. Okay. So basically, just to recap, we've got really sort of two main analyses that are being used in the field now, sort of DFC windowed estimates, clustering, figuring states from that, and then going directly from the data, hidden Markov modeling, and coming up with those states. And you're going to get different answers. And so I think it's up to us to figure out what is the right way to proceed, and essentially what is actually potentially even the connection between these two methods. Okay, so now I'm going to get on to sort of the last two topics I'm going to cover are sort of reasons why you might be not so enthusiastic about doing DFC estimates, so what problems you might face in doing DFC estimates, and are the DFC estimates really telling us something about the brain that we care about, or are they telling us something else? So one thing I think, especially with resting state studies, that's becoming more and more obvious and was mentioned in one of the member-initiated symposiums was the effects of vigilance and wakefulness. Obviously, for those of you who've been in subjects in the scanner, it's pretty hard to sort of stay awake in a resting state scan or to even maintain any reasonable state. And so in this study, what they're looking at is they are finding, so these are different states, connectivity states that they've identified from the bold time series. And here, they also had EEG, and so they also knew whether they were awake or N1, N2, or N3 stage of sleep. And you can sort of start seeing here, there is some correspondence between states and wakefulness, okay? And this is state C1, and the subject's fairly awake here. In C2, the subject appears to be here in N1 a little more, okay? And so what they did is they quantified that, and essentially you could do k-means clustering to come up with your states, or you could cluster the data based on what sleep state they're in, and you can sort of see these look very similar, okay? And one sense might be that the states that people are finding are essentially we're just measuring people's wakefulness state. And so, for example, in that previous example I gave you with healthy controls versus schizophrenia, it may just be that the schizophrenics were hanging out in a different vigilance state than the healthy controls, okay? And in which case, there's really no reason to do fMRI. You can just look at their sleep states. The last thing I'm going to talk about is global signal and nuisance regression. So everyone who does fMRI you've probably done nuisance regression, which is essentially there's a lot of stuff in the signal that we think is not what we want to measure. And so, for example, this is a nice study from Power and others. This is sort of a plot of the data where every row is a voxel and across here is time. And you can sort of see there's these very global sort of signal variations here. And these correspond to, for example, either in this case, you're seeing a lot of sort of respiration and heart rate based sort of correlation. So as you see these heart rates, these respiration activity is changing, you're seeing a lot of changes in the fMRI signal. One thing that people often do is they just average the signal to come up with a global signal. And so another option is you could just regress that global signal out of this data to help remove some of these effects, okay? So it has been a long tradition in fMRI to try to remove what are called nuisance effects. So I just want to talk a little bit about that. So for resting state fMRI, by far the most common method is with something called global signal regression, which is, I would say, used by at least half the studies that have been published, and yet is very controversial. There's many papers arguing that it's not the right thing to do, and yet people do it because it actually really cleans up your data really well, okay? And I think there's arguments on both sides for and against global signal regression. So I'm not here to solve that controversy, but just to sort of point out that it is something that is commonly done for traditional static functional connectivity, and therefore we would expect it to also have similar issues when we're applying it to dynamic functional connectivity. So for example, in this study from NeuroImage that was just published, these are some of the correlations, windowed correlations before global signal regression, and these are the global signal regressions after. And you can sort of see for some windows, there's almost no effect of global signal regression, and other windows, there's quite a large effect, okay? And so the question is, what is the right thing to do? And so that's still open for discussion, but the bottom line is whether you do it or not is going to have a huge impact on what you think the states of the brain are. So if you don't do global signal regression, these are going to be the states you're going to estimate. And if you do global signal regression, then these will be the states that you estimate, and you'll end up with different dwell times and different interpretations. So I think that this is something that needs to be considered. And just as an aside, that the global signal, there is evidence showing that global signal is also related to vigilance fluctuations. And so once again, it may just be that global signal regression is sort of removing some of the vigilance fluctuations and giving you states that are less maybe prone to wakefulness. And as you can see, if you do that, then the states look a lot more similar to each other. Whereas here, when you leave in the vigilance fluctuations, there's a little more variation across states. Okay? So I think this is something that the field needs to consider a little bit more. So my last slide before I summarize is just talking about an issue that we've been looking at and that was discussed in an abstract yesterday, which is just the fact that what we found is, so in this case, what I'm showing you here is in blue is the dynamic functional connectivity. So in each of these, we've estimated the dynamic functional connectivity across time before we've done nuisance regression, okay? And then in black, what we've done is we've plotted the magnitude of a nuisance term. So for example, this is the white matter and CSF nuisance term, this is global signal, and this is head motion. And you can see and probably not surprisingly, there is a fairly strong correlation, or there can be, between the DFC estimate in blue and the black, which is a nuisance magnitude, okay? So that's probably not too surprising. If there's a nuisance term in your data, it's probably going to get into your DFC estimate. The bad news is typically what we do is we regress that signal out using general linear models, and then we think we're done, which is basically we've gotten rid of the nuisance term. What we found, though, is in red here, we're showing the DFC estimate after you've done nuisance regression. And as you can see, we really haven't removed the relationship between the nuisance term and the dynamic functional connectivity estimate. There's sort of a whole theory behind this, but for now, I'm just going to point this out as something I think as a field we probably need to think about a little more, is that, all these things we're measuring, to what extent are they related to these nuisance terms or vigilance? And there is no sort of separation in the sense that, one can argue that head motion and vigilance are quite highly correlated. Global signal and vigilance are highly correlated. So all these things are sort of part of the same picture in the sense that, there are nuisance terms, but ultimately at the end of the day, it's the brain that's controlling your breathing, your cardiac, your movement. And I think as a field, we need to start thinking about disentangling these factors in a better way in terms of the distinction between nuisance and signal is not necessarily that clear. So to summarize, most DFC studies have been based on sliding window approaches. And then from these sliding window estimates, you can estimate DFC states. You can look at some of the properties of these states, such as transition probabilities and dwell times. But there is growing interest in more direct data modeling, and these approaches can address some of the issues, including the idea that you don't need to prescribe any fixed window length. And so I think more work needs to be done to compare these two different approaches. I think there's got to be a better, as a field, we probably need to do a better job of taking into account arousal and vigilance variations as a potential source of DFC fluctuations, and this might suggest additional measures, either EEG or monitoring the eyes or other physiological measures. And I think in general, nuisance regression has not really been discussed much in the DFC field, but I think it definitely requires more attention because I think there is growing evidence that a lot of these fluctuations may be driven by factors that may not be what we think they are, may not be the factors of interest per se. So what I've included in these slides is also some suggested reading. There are preliminary slides on the website, and I've updated these slides since then. So if you want a copy of these most recent slides, just send me an email. I'd be happy to share a link with you. So thanks for your attention, and I'm happy to take questions. Thanks.